My name's Catherine, and I'm going to be taking you through our genetic testing process today. So, you have kindly provided us with a sample of your DNA in the form of your saliva. And what we're going to do is we're going to isolate that DNA from your cheek cells, many of which will be in the saliva. And after we've done that, we're going to be able to tell you all sorts of things about your ancestry, so where your relatives came from in the past. We can link you to people who you're related to. We can tell you what you're genetically predisposed to, but really these should be treated as just interesting things about yourself. They're not anything to base any real decisions off, or to decide where you're strong and where you're weak, because as we know, how we're raised, how we live our everyday lives, our lifestyle choices, they all play a huge role in deciding which attributes we have. For example, there are professional athletes without any predisposition genetically that's associated with athleticism, and the same goes the other way around. So you might not be genetically predisposed to have a certain health problem, but you may get it anyway due to chance and lifestyle choices. So when it comes to things like that, it's really just trivia about yourself. So the first thing we're going to do is we're going to trace your ancestry. So most genetic testing companies, they do one type of ancestry testing where they find out where your ancestors lived in, but we go a step further, or rather 2 steps further, because they typically analyse the DNA on your chromosomes, excluding your sex chromosomes. We'll get to that later. But your chromosomal DNA, called autosomal DNA, that's the DNA that mixes between your father and your mother when they copulate and produced you. Because all of that DNA mixes together, you can only trace back 5 to 8 generations, which means 200 to 300 years, depending on how young or old the parents of each generation were. So most genetic testing companies, when they provide, the regions that you came from, it will be based on more recent generations of your ancestry. However, there are 2 types of DNA that you can use to trace your ancestry back tens of thousands of years. So the first one of these is the DNA that you find in your mitochondria. And just to recap your knowledge on the mitochondria, they're the little parts of the cell which are responsible for generating energy so that your cells can do pretty much anything. So if you move, if you think, if you're carrying out pretty much any chemical process in your body, if you're digesting, it's all using lots of energy, and mitochondria are the little tools within the cell that provide that energy. So mitochondria has its own DNA, and this is passed directly from mother to offspring. So to just provide a little aesthetic aid, if you see, it goes directly from mother to offspring to offspring to offspring, and it would be completely the same if this here, I've been very gendered and I've put a male child as blue and a female child as red. So it looks like it's just going from mother to daughter to daughter to daughter. And then here at the bottom, whether you're male or female, the DNA within your mitochondria is from your mother. So if you are a man, your children would not get your mother's mitochondrial DNA, but if you are a man, you have your mother's mitochondrial DNA, if that makes sense. In the same way that whether you have a male or female child, they will get their mother's mitochondrial DNA. We have a direct line down the maternal line, as in from mother to mother to mother, tens of thousands of years. And because the DNA isn't recombined and it stays pretty much the same over this entire time, this means that we can trace our ancestry much further back. All living humans today have a common human ancestor that originated in East Africa. And that was around 200,000 years ago, I think. And then this ancestor had the original mitochondrial DNA, shall we say. But then over tens of thousands of years, the humans descended from this ancestor. They spread out all over Africa. And within this amount of time, tiny changes can happen to mitochondrial DNA where there's just one mutation in the DNA over thousands of years. So it remains pretty much the same regardless of who you're looking at in the world. We all have pretty similar mitochondrial DNA sequences, but these tiny changes are what allow us to see our ancestry, because it pretty much follows the migration patterns of human beings over tens of thousands of years. So it started out in East Africa with a particular mitochondrial DNA pattern, and we would refer to these as haplogroups. And the haplogroup just refers to the region where your particular mitochondrial DNA is linked to, and also the pattern of the DNA. So the whole thing is just the haplogroup. It's the mitochondrial DNA group you belong to. So there were a couple of groups that arose in Africa, and there was L0, L1, L2 and L3. These letters don't really mean anything, they just refer to slightly different variations of the DNA. So then a group of humans migrated out of Africa. So all non-African people are descended from the L3 group that originally came from Africa. So L3 moved out into the Middle East and we think South Asia, and then by that point tens of thousands of years are passing, and more genetic mutations are happening. So when there are just these tiny little mutations that are happening, it's also kind of tracking the migration and settlement patterns of people at that time. So we find different haplogroups to be very common in certain areas, but essentially where people have migrated and settled. So there'll just be one person where a mutation has happened, and then all of that person's descendants, that female's descendants, will have that mitochondrial DNA pattern, and then it just gets spread over that particular population. There is quite a bit of mixing of different haplogroups in the sense that the descendants of a person who acquires this mutation passes on the mutation. It's not like people near that person at the time can have it, so you'll still have the L3 haplogroup also in those areas, but new haplogroups emerged where they had lots of descendants, and so it made that haplogroup quite popular in that area. And then there was migration to Europe, Asia, the Americas, and the same thing happened where at some point a mutation will have happened, and they had a lot of descendants who all carried that mutation. And again, there'll still be a variation of different haplogroups within those areas. And because of the high frequency or the total absence of haplogroups in certain areas, we can pretty much say where your ancestors from tens of thousands of years came from, because obviously way back then the human population was very small, so it was quite common for just haplogroups to just completely dominate a particular region of the world. As I said, the L2, L1 and L0 haplogroups only existed in Africa, and then L3 moved out, and then a couple of other haplogroups dominated other parts of the world from that point. So that's the mitochondrial DNA, so we can trace it directly down the maternal line and figure out where your maternal tens and tens of thousands of years ago. And we can do a very similar thing with the Y chromosome DNA. So with the Y chromosome DNA, I will get my visual aid again. It's exactly the same thing, where it goes from male to male to male to male. And then a difference here is that it can only go to a male also. So if a woman wanted to trace her paternal lineage, she would be able to get a DNA sample from her brother or from her father, and then that would tell her where her paternal DNA came from. Because females do not have a Y chromosome. We have XX sex chromosomes and males have XY chromosomes. It's almost exactly the same as the mitochondrial DNA in that it doesn't recombine, apart from just 5% of the Y chromosome DNA recombines with the X chromosome DNA when the egg and sperm fertilize. But it's the same 5%. It's not like a random 5% of each Y chromosome is switched, because then again you'd have the issue where it's just all recombined and you wouldn't be able to trace back very far. But it's just this one section that is recombined, so we just ignore that section and we look at the 95% that does not recombine and stays as the mitochondrial DNA does. Almost exactly the same over many thousands of years, and you can find where your paternal ancestor is from, going from male to male to male to male over tens and thousands of years. And they have their own haplogroups, and they have a different naming system, a slightly more intuitive naming system that goes A, B, C, D, E, F, rather than starting at L and going all over the alphabet, basically, which is what happens with mitochondrial DNA. So that is how we will determine your ancestry from the Y chromosome DNA and from the autosomal or just normal chromosome DNA. And the normal chromosome DNA, I'll explain this briefly, it's just there'll be certain patterns in recombined DNA over 5 to 8 generations that we will still be able to link to a particular place. But yeah, after that we can't do that anymore. So I think it's good to have all of the information. I think personally that the tens of thousands of years ancestry is just as interesting, if not more, than finding out your more recent relatives. So the next thing we do is we search for your potential relatives that we might have in our database, and we do this by looking at your IBD segments of your autosomal DNA, your chromosome DNA, and these are sections that you will share with relatives if you are related. So if you share a common ancestor like a great-grandmother or a great-great-grandmother on our database, then we will be able to link you to them. So whether they're your third cousin or any other kind of relative with whom you share a relatively recent living ancestor, then we will be able to link you up to them. Now we'll also be able to tell you certain things about what health risks you might be predisposed to, but as I mentioned earlier, this should very much be taken with a grain of salt, because you can be predisposed to something and not get it, or you can not have anything flag up on our system, but you can still get a condition. For example, almost every single person, apart from people with very, very specific conditions, will be able to get obese even if you do not have a genetic predisposition for becoming obese. Likewise, it's important to note that if we do say you have a predisposition to something, it doesn't mean that you're going to get it. It just means that statistically, people with the genetic marker that you have are found to have a condition in a higher frequency than the rest of the population. So you can bear in mind health risks if you want to, if that's going to help you make healthy lifestyle choices, or go for checkups more regularly. I think that's a good thing. I think actually the more harmful thing can even be being told that you have no genetic predisposition to something, so you just think you're invulnerable and you end up doing harm to your health, and then you get health conditions regardless, because we all know how important lifestyle is, and for most things lifestyle is more important than genetics, in health especially. There are obviously plenty of exceptions to that rule, but for the most part, the things that we're looking for, the molecular genetic markers, are called single nucleotide polymorphisms. This essentially just means, so imagine this is a DNA sequence, and so that's the original sequence. Obviously single is just one of these, and then each one of these letters represents a nucleotide within your DNA sequence, and that is what your DNA sequence is made up of. And polymorphism, you can essentially just think of it as a change. So what would happen is this here would be a single nucleotide polymorphism, and these are associated with all kinds of things, whether it's your ancestry or your predispositions to have certain attributes. So those are particularly useful for working out the haplogroup of your ancestry, because it's these tiny changes that are happening over time and in specific regions, and which kind of track the migration patterns over time. They're not massive changes, it's just literally a single nucleotide change to the DNA sequence, so just a single letter. Not always a single letter in terms of it doesn't have to be one mutation that characterises a new group. There could be a couple of mutations that make one group different from another, but generally it's not huge changes of the DNA. It'll be one change at a time. It's important to note that with all of these kinds of tests, there are false positives that can happen. We try and be as accurate as possible, but mistakes do happen. And I also feel like it's my duty to tell you, or just remind you, that genetics have a very variable role in all sorts of things. Like, our attributes are about training them. You might have a genetic predisposition to something, in terms of that will make you good at it, but there are plenty of people who have no genetic predisposition for a certain thing, and they go on to be highly successful in that field. And it's also important to note that test the genes that we know about that contribute to these things, but our knowledge is still very much limited in terms of how genes interact with each other, the environment, all of the genes that might contribute to a particular skill in someone. I mean, something like, for example, certain sports have lots of skills involved, for example, like muscle strength, but also hand-eye coordination and just quick thinking, stuff like that. Like, all of the different combinations of genes that have such a complex effect on, the phenotype of the individual. That is what is displayed in the individual. So really, for things like attributes and what your strengths and weaknesses might be, you should just judge that based off your own perception. And even that should be challenged at times, because it's my belief, and there have been plenty of studies done that support this, that the most important thing you can do is just work hard at something, put a lot of time into something, and focus if you want to be very, very good at it. And that will override genetics in 90% of circumstances. Obviously, there are people with severe learning disabilities that I'm not going to state that absolutely everyone could become the world's best mathematician, but I do think in general we're a lot more capable of doing whatever we would genuinely like to do, as long as we're willing to put in the hours to train our brain or train our muscles to do it, essentially. So yes, that's my little philosophical thing that I'm going to pull away from now, because I'm meant to be just talking to you about our genetic testing process. So do you have any questions for me? Good question. So we extract the DNA from the cheek cells in your saliva by, well, we have lots of chemical processes and a few different methods that we use to isolate the DNA, get rid of all the other cell contents, so get rid of the proteins, get rid of the membranes, get rid of everything from the cells and from the sample that is not DNA. We isolate that DNA and then we sequence it using one of a few methods, and how that's done is we work out the sequence of nucleotides, those little letters that I showed you, in your entire DNA sequence, at least the parts that we're looking at. And we do this by essentially, we use these things that can bond to one of the nucleotides and doesn't bind to any of the others, so we know which little nucleotide is in the sequence based on which one of our little, we'll call them detectors, have bonded to it. And we can fluorescently label these, so they'll come up on the machine as a particular colour, for example. There are different ways to do this, but that's the one that we use. And from that, it sounds very slow, but the machine can do it very, very quickly. So it just trial and errors binding to those nucleotides, and based on what the fluorescent labels tell us, we can correspond that so that we can write out the whole sequence, and that's how we do it. Okay, well, it's been fantastic to talk with you. It's not often that one of our clients actually, wants to hear about the entire process, and I'm surprised you haven't walked out on me, so I'm very glad and pleased and grateful that you're still here. And if you're confused about anything, or if you have any follow-up questions, just let me know.